Hello, everyone. My name is Marcos Matabuena, and I am a professor of biostatistics and digital epidemiology. In this video, I will introduce my new paper reading course called Digital Biostatistics. Throughout the course, we will discuss 16 papers organized into 5 thematic blocks that, in my opinion, cover some of the central statistical challenges that arise when we work with digital health data. The overall objective is to understand how quasi-continuous data collected from wearables, smartphones, continuous glucose monitors, and other digital devices can be transformed into useful information to better characterize human health and ultimately improve clinical decision-making. In the first block, we will study how to construct appropriate statistical representations of digital health data, instead of summarizing thousands of observations. Using only a mean or a few conventional features, we will study functions, probability distributions, and multi-level structures that preserve much more of the underlying physiological information. In the second block, we will focus on latent structure and digital phenotyping. Here, the goal is to discover patterns that are not directly observed in the data subgroups of individuals, latent physiological states, or disease trajectories that may help us better understand human health. In the third block, we will move to high-dimensional biomedical discovery. We will examine problems involving thousands or even millions of variables, as is common in genomics and transcriptomics, and discuss methods such as the lasso genome-wide association analysis and by clustering. An essential part of our digital biostatistics agenda is not only making predictions but also understanding how uncertain those predictions are. Therefore, the fourth block is devoted to predictive uncertainty and conformal prediction, including the more challenging situation in which the outcome is not a number but a function, a distribution, a trajectory, or another non-Euclidean object. Finally, in the fifth block, we will return to raw digital health data. We will discuss missing data in GPS and mobile sensing, how to construct reliable digital biomarkers from incomplete observations, and how to connect digital measurements with precision medicine and individualized decision making. So, the overall journey of the course is from sensing to representation, modeling, phenotyping, discovery, uncertainty, and finally, decision. I hope you enjoy this course, which brings together statistical ideas that I believe are essential for understanding digital health data and taking healthcare into a new dimension. Let us now explore the course block by block. We begin with the first block, statistical representations of digital health data. This block is fundamental because before fitting a machine learning model or performing statistical inference, we need to answer a much more basic question. What is the appropriate statistical representation of an individual? A wearable device may generate thousands of measurements per person. A continuous glucose monitor records glucose every few minutes. An accelerometer continuously measures physical activity. If all of this information is reduced to a mean, a maximum, or a small number of summary statistics, a large part of the underlying physiological structure may be lost. The objective of this block is therefore to introduce statistical representations that preserve the richness of continuously measured health data. We will move from probability distributions to functional data, repeated functional observations, and finally, multi-level distribution valued outcomes. This block provides the mathematical language that we will use throughout the rest of the course. The first paper is glucodensities, a new representation of glucose profiles using distributional data analysis. The main objective of this paper is to propose a new statistical representation of continuous glucose monetary data. Traditionally, a glucose profile is summarized using variables such as mean glucose time in range, glucose variability, or other scalar CGM metrics. The problem is that these summaries compress thousands of glucose measurements into only a few numbers. In this paper, we take a different approach and represent the complete glucose profile as a probability distribution, which we call the glucodensity, instead of describing a person only by their average glucose. We retain information about the complete amount of time that the individual spends across different regions of the glucose distribution. Why is this paper important for the course? Because it introduces one of the central ideas of digital biostatistics. The statistical representation of the data is itself part of the scientific problem. Before selecting an algorithm, we should ask which mathematical object best preserves the physiological information contained in the digital signal. The second paper is glucodensity functional profiles outperform traditional continuous glucose monitoring metrics. After introducing a richer representation of CGM data, the natural next question is whether that representation actually provides additional clinically relevant information. That is the main objective of this paper. We compare glucodensity functional profiles with conventional continuous glucose monitoring metrics and investigate their ability to characterize metabolic differences and predict future health outcomes. Two individuals can have almost identical mean glucose or time in range while having very different glucose distributions. By using the complete distributional profile, we can capture metabolic patterns that remain invisible when we use only conventional scalar summaries. Why is this paper relevant? Because it connects statistical representation with clinical prediction. A richer representation is not useful only because it is mathematically elegant. It is useful if it preserves additional physiological information that can ultimately improve digital biomarkers and predictive algorithms. The third paper is the classic multi-level functional principal component analysis. Here, we introduce another major characteristic of digital health data: repeated observations from the same individual. For example, we may observe several days of physical activity, multiple nights of sleep, or different periods of continuous glucose monitoring for the same person. The objective of this paper is to extend functional principal component analysis to separate different sources of variability. In particular, it distinguishes between subject variability, which describes how individuals differ from one another, from within subject variability, which describes how repeated observations from the same person change over time. This distinction is essential. If one person provides 7 days of wearable data, those 7 days are not 7 independent individuals. Why is this paper relevant to digital biostatistics? Because wearable and mobile health data are intrinsically multi-level and repeated measures data. Understanding this hierarchy is critical for studying physiological variability and building personalized models. The fourth paper is fast univariate inference for longitudinal functional models. Once we recognize that digital health data are functional and longitudinal, another challenge immediately appears: computational scale. We may have thousands of individuals, multiple days per individual, and hundreds or thousands of measurements within every day. The objective of this paper is to provide a computationally efficient framework for inference in large longitudinal functional datasets. The strategy is particularly elegant. Instead of fitting one extremely complex functional model directly, the method fits univariate mixed models across the functional domain and subsequently smooths the resulting estimates. This makes it possible to estimate functional effects under uncertainty in a scalable manner. Why is this paper important? Because digital biostatistics methods must not only be statistically valid, they must also be computationally feasible for the size and resolution of modern wearable datasets. This paper introduces another central idea of the course: statistical scalability. The final paper in this first block is multi-level functional distributional models with applications to continuous glucose monitoring in diabetes clinical trials. This paper combines several of the ideas introduced in the previous readings. The objective is to analyze situations in which each individual has repeated longitudinal observations and each observation is itself represented by a probability distribution. Imagine, for example, a diabetes clinical trial in which participants wear a continuous glucose monitor at baseline and again at several follow-up visits, instead of asking only whether mean glucose changes after treatment. We can study how the entire glucose distribution changes over time. The proposed framework allows us to separate different levels of variability while simultaneously studying treatment or longitudinal effects. Why is this relevant? Because it illustrates the kind of statistical problem that arises when digital biostatistics is applied directly to clinical research. We move from analyzing individual digital signals to modeling repeated multi-level and non-Euclidean outcomes. The second block is devoted to latent structure and digital phenotyping. Once we have appropriate representations of digital health data, next question is, can we discover structures that we did not define in advance? In medicine, we often begin with predefined categories, such as healthy versus diseased, responder versus non-responder, or diabetes versus no diabetes. But human physiology is likely to be much more heterogeneous. Rich longitudinal data may reveal previously unknown subgroup, physiological states, or disease trajectories. The objective of this block is to introduce different statistical paradigms for discovering this hidden structure. We begin with the EM algorithm, one of the foundations of latent variable modeling. We then study multi-level mixture models, kernel-based clustering, and finally a modern framework integrating longitudinal chronic health records with genetic information. The overall objective is to understand how we can move from digital measurements to latent digital phenotypes. We begin with one of the great classics of modern statistics, maximum likelihood from incomplete data via the EM algorithm by Dempster, Laird, and Rubin. The objective of the paper is to develop a general procedure for maximal likelihood estimation when part of the relevant information is not directly observed. The algorithm alternates between two steps. In the E step, the current parameters are used to estimate the missing or latent information. In the M step, that information is used to update the model parameters. The process is repeated until convergence. Why are we studying a paper from 1977 in a digital biostatistics course? Because a remarkable number of modern statistical models are conceptually connected to this idea. Mixture models, latent class models, hidden state models, random effects models, and many generative approaches. The EM algorithm therefore provides an important mathematical foundation for the digital phenotyping methods that follow. The next paper is mixture models with multiple levels with application to the analysis of multi-factor gene expression data. The objective is to extend traditional mixture models to situations in which latent structure may occur at several different levels. In a standard mixture model, we might imagine that the population contains several unknown groups, but biological data can contain much richer hierarchical structure, major groups, subgroups within those groups, and potentially different molecular processes acting simultaneously. This paper develops models that can capture this multi-level heterogeneity and applies them to gene expression data. Why is it relevant to digital biostatistics? Because digital phenotypes are also unlikely to exist at only one level. We may observe groups of individuals with similar physiological patterns while simultaneously observing different temporal or biological structures within those groups. This paper encourages us to think about phenotyping as a hierarchical and multi-level problem, rather than simply applying a clustering algorithm and selecting a number of clusters. Paper number 8 is kernel K groups via Hartigan's method. Until now, we have considered clustering mainly from a probabilistic model-based perspective. This paper introduces a different approach. Its objective is to develop clustering methods based on energy statistics, distances, and kernels, without necessarily assuming a specific parametric distribution for the data. Kernel methods also make it possible to identify nonlinear cluster structures and work with much more flexible representations. Why is this particularly relevant in this course? Because many digital health objects do not naturally belong to ordinary Euclidean space. They may be functions, probability distributions, trajectories, or other complex objects. If we can define an appropriate distance between these objects, we can build clustering algorithms directly on their geometry. This paper therefore connects digital phenotyping with the non-Euclidean representations introduced in the first block. We close the second block with a very recent paper, a Bayesian framework for longitudinal EHR and genetic discovery. This work illustrates how far latent structure methods can be taken when they are combined with very large biomedical data. The objective is to integrate longitudinal electronic health records and genetic information in order to identify latent disease signatures that evolve over time, rather than describing a disease using only a binary diagnosis. The framework learns dynamic patterns from the complete medical history of an individual and subsequently connects these patterns with genetic variation. Why is this paper particularly important? Because it demonstrates a major change in perspective. Longitudinal data can be used not only to predict disease, but also to redefine the phenotype itself before performing genetic discovery. In my opinion, this integration of longitudinal phenotyping and genomics represents one of the most exciting future directions for digital biostatistics. We now enter the third block, high-dimensional biomedical discovery. Once we have created meaningful digital phenotypes, we can ask which biological variables or mechanisms are associated with them. This introduces a characteristic problem of modern biomedical science. We may have many more variables than individuals. A genetic study can include millions of variants. A transcriptomic experiment can contain tens of thousands of genes. The objective of this block is to study how statistical discovery can be performed in these high-dimensional settings. We begin with the lasso, one of the fundamental methods for regularization and variable selection. We then apply similar ideas to genome-wide association studies. Finally, we study by clustering, where the objective is no longer simply to select variables, but to discover joint structures involving both subsets of subjects and subsets of biological features. The first paper in this block is one of the most influential articles in modern statistics, Regression Shrinkage and Selection via the Lasso by Robert Tibshorani. The objective of the paper is to address two problems simultaneously: building an effective predictive model and selecting a relatively small number of relevant variables. The lasso introduces an L1 penalty on the regression coefficients. This penalty produces shrinkage, but it has another particularly important property. Some coefficients can become exactly zero. The resulting models are therefore sparse. Why is the lasso relevant to digital biostatistics? Because many biomedical problems are intrinsically high-dimensional. We may have thousands of digital biomarkers, genetic variants, image-derived features, or variables obtained from multiple data modalities. The lasso introduces a fundamental principle for these settings. Regularization allows us to control model complexity while performing variable selection. The next paper is Genome-Wide Association Classes by Lasso Penalized Logistic Regression. Its objective is to bring the Lasso framework into the context of genome-wide association studies. In a US, we may have hundreds of thousands or millions of SNPs, but a much smaller number of individuals. This is a classic situation where P is much larger than N. The paper uses penalized logistic regression to identify a sparse collection of genetic variants associated with a phenotype and also explores potential interactions among variants. Why is this paper relevant? Because it provides a direct connection between high-dimensional statistical methodology and biological discovery. It also gives us an opportunity to discuss an extremely important distinction. A variable selected because it improves prediction is not automatically a causal biological mechanism. So this paper allows us to distinguish between prediction, association, variable selection, and scientific discovery. The final paper in these blocks is Big Clustering Algorithms for Biological Data Analysis, a survey. Traditional clustering usually groups individuals using all available variables or groups variables using all available individuals. But biological structure may exist only in part of the data matrix. For example, a particular subset of genes may exhibit a meaningful pattern only within a specific subset of patients. The objective of big clustering is to discover both of these structures simultaneously. This survey reviews major approaches to big clustering, particularly in gene expression analysis. Why is this relevant? Because it introduces an important idea for heterogeneous and multimodal biomedical data. The relevant signal may be local rather than global. Not every biomarker needs to be informative for every individual. This perspective naturally connects with precision medicine, where different biological mechanisms may be important for different patient subgroups. The fourth block is devoted to predictive uncertainty and reliable AI. Until this point, we have discussed representation, modeling, clustering, discovery, and prediction. But in medicine, a prediction without information about its uncertainty can be extremely difficult to interpret and potentially dangerous. Therefore, one of the central questions of digital biostatistics is how certain are our predictions? The objective of this block is to introduce methods that accompany a prediction with a region or set that formally quantifies uncertainty. We begin with conformal prediction in the classical setting. We then extend the problem to much more complex responses, such as probability distributions, functions, trajectories, graphs, and other objects living in general metric spaces. The first paper is a tutorial on conformal prediction by Glenn Schaefer and Vladimir Vovk. The main idea behind conformal prediction is conceptually simple. Instead of producing only a point prediction, we construct a prediction set that contains a future observation with a controlled probability. One of the most attractive properties of conformal prediction is that these guarantees require relatively weak assumptions, principally exchangeability. Conformal prediction can also be wrapped around almost any predictive algorithm. We may train a random forest, a neural network, or another machine learning model, and then use a calibration sample to quantify predictive uncertainty. Why is this important for digital biostatistics? Because it allows us to separate two problems. First, we construct the best predictor we can. Then we place a layer of statistical reliability around that predictor. This separation between prediction and uncertainty quantification is particularly important for clinical applications. The second paper in this block is conformal and KNN, predictive uncertainty quantification algorithms in metric spaces. Here we return directly to the ideas introduced at the beginning of the course. In standard conformal prediction, the outcome is usually a scalar or a vector. But what happens if we want to predict a probability distribution, a function, a graph, or an entire trajectory? The objective of this paper is to develop predictive uncertainty quantification methods when the response belongs to a general metric space. The work studies conformal procedures and nearest neighbor approaches for constructing predictive regions for these complex objects, including locally adaptive methods for heterogeneous data. Why is this paper relevant? Because it closes the loop between representation and uncertainty. If we choose to represent an individual using non-Euclidean statistical objects, we also need inferential tools that respect that geometry. Otherwise, we would build a rich representation only to reduce it again to a scalar because our classical statistical tools could not handle it. We now reach the final block of the course, from digital biomarkers to precision medicine. Until now, we have often behaved as if digital health data were already available in a clean and complete form. In practice, this is rarely true. Smartphones and wearable devices produce imperfect data. Sensors may stop recording. Users may remove their devices. GPS signals may disappear, and missingness itself can be related to the behavior of the individual. The first objective of this block is therefore to understand how we can transform raw and incomplete sensing data into reliable digital biomarkers. But we then want to take one final step. The goal of digital biostatistics should not only be to describe or predict health. Ultimately, we want to use this information to improve decision making. For this reason, we finished the course by introducing dynamic treatment regimes and examining how longitudinal digital information can be transformed into individualized clinical decisions. The first paper in the final block is Informing Mobility Measures from GPS Traces with Missing Data by Ian Barnett and Jukka-Pekka Anela. At first sight, the problem seems simple. Use smartphone GPS data to measure human mobility, but real GPS data are highly incomplete. The phone may turn off GPS to conserve battery. Signals may be unavailable, and only fragments of an individual's actual trajectory may be recorded. The objective of this paper is to develop statistical methods that reconstruct mobility characteristics from these partial observations and allow us to derive measures such as distance traveled, time spent in different locations, or other mobility indicators. Why is this paper important? Because it demonstrates a fundamental idea. A digital biomarker is not simply a variable that we download from a device. There is an entire statistical process between the raw signal and the health characteristic that we ultimately analyze. If we ignore the observation mechanism or the missing data process, the resulting digital biomarkers may be seriously biased. And we arrive at the final paper of the course, estimating dynamic treatment regimes in mobile health using e-learning. In many ways, this paper represents the final destination of the entire course. Its objective is to learn dynamic treatment regimes, rules that use the current state of a patient to determine which intervention should be recommended at a particular moment. In mobile health, the state of an individual can be continuously updated using information about physical activity, sleep, glucose, behavior, and other digital signals. B-learning uses ideas related to reinforcement learning to learn a policy that transforms this longitudinal information into individualized treatment decisions. The paper is also motivated by applications in diabetes. Why have we chosen this as the final paper? Because it transforms digital biostatistics from a discipline focused only on observation and prediction into a discipline focused on decision making. We began the course by asking how to represent thousands of measurements produced by a digital device. We finished by asking a much more ambitious question. Given everything we know about this individual right now, what should we do next? In my opinion, this transition from sensing to individualized decisions captures the ultimate objective of digital biostatistics.